Okay, thank you so much for the introduction, and thank you so much for the opportunity to present today. I'm Ati, 40-year PhD candidate from University of Waterloo, and today I'm going to talk about calibration of agent-based model against microscopy data. Okay, I want to start my talk with this question: why do we even care about engineering microbial communities? And the very immediate answer to this type of question is because in this type of engineering supports a wide range of application, from human and animal health to sustainable agriculture to wastewater treatment and so on. For example, we could install a gene in ocean microbiome to produce an enzyme to degrade microplastics. Or we could install a gene in gut microbiome to produce, let's say, lactase to help lactose intolerant people consume dairy products. Or we could install a gene in soil microbiome to augment nitrogen fixation to help fertilizing crops. But there is a challenge here. And the challenge is bacteria are notoriously hard to engineer predictably. And there are two main reasons here: spatial heterogeneity and microbial community heterogeneity. I'm not going to talk about community heterogeneity today. I'm going to leave it for folks in metagenomics. And instead, I'm going to talk about spatial heterogeneity. What does it mean? Spatial heterogeneity basically is where individual cells encounter vastly different microenvironments. For example, in this slide, you can look at gut villi in blue that is colonized by a patch of one strain in orange. And you can look at soil pores that concentrate bacteria along its grain surface. In both studies, they show that basically spatial heterogeneity governs microbial function. And what we can learn from these type of studies is basically such fine scale heterogeneity prevents prediction of basically community response and engineering basically these type of communities. So what we need here, how we can address this challenge, is basically we need a framework to be able to capture single cell, individual cell responses. This requirement led the community to use of agent-based models. Agent-based models are also known as individual-based models in the field of ecology, is where each bacterium is represented as an agent in its local chemical landscapes. And here we are looking at one representative of agent-based model. And in this specific study, basically this is a synthetic consortium. There are two bacteria involved, and they are communicating via current sensing. And the authors had to use agent-based model because they needed to treat each bacterium as an agent to be able to capture the signals that they are giving and receiving. And the result of this study was also very striking because they found that even if you keep all of the parameters the same, even if the genetic circuit is the same, only by rearranging the spatial context, you're going to get different outcome. So spatial context can make or break a design. We as systems biologists lean on use of agent-based model, not just because they are able to reproduce observed behavior, but also because they are able to generalize across conditions, predict unseen behaviors, and ultimately guide rational engineering of microbial communities. This is where our famous design, build, test, learn cycle comes in. And models like agent-based model are central to the design section. This is where you can basically explore over your parameter space, prioritize your experiment. But today, we're not going to do any design. Instead, we're going to focus on learn section because we're going to learn from our model. We're going to calibrate our model. We're going to understand how we can fetch mechanistic insight from our agent-based model to hopefully inform the next cycle of design. The pipeline I'm pitching today starts with, basically has two different phases: experimental phase and computational phase. Our experimental phase starts with time-lapse microscopy. I have to acknowledge that I understand that the very first step is different across different groups based on the question that they try to answer. But our group has time-lapse microscopy to be able to capture bacterial interaction at the single cell level. And then we want to be able to basically transform our images, our time lapses, to numbers This is where image processing comes in. And then we're going to integrate our experimental phase to our computational phase through a sequence of statistical pattern extraction. We call these summer statistics, but they are essentially patterns. And then we have our model simulation. I want to emphasize that our models are agent-based model, and then we have parameter inference. This slide is going to serve as a roadmap for the rest of my talk, and I'm going to walk you briefly in each step. Okay, the first step, collection of spatial-temporal data. So we do have access to do two different setups in our lab, AgarPad and microfluidic device. So AgarPad is very straightforward to set up, but it's going to give us basically less control over nutrient flow. In contrast, microfluidic chip basically going to give us this access to have more control over the flow. So basically we can go for longer time lapses. And to kind of give you an idea of how long, sometimes my experiments are 72 hours. And our microfluidic chip is designed and built in collaboration with Matt Courtney from Microfluidic Lab at U Waterloo. Okay, you have your data. Once your spatial temporal data is collected, you need to basically transform these data to structured information like shape, orientation, longitudinal information. And there are lots of lovely software available online, but we saw the need to benchmark our pipeline for image processing. And basically this slide is our benchmark pipeline. So we start with segmentation. And for the segmentation, we use the unit-based algorithm called Omnipose. And then we pass this segmented mask to Cell Profiler. And Cell Profiler does the job of tracking for us. Basically, it links objects or cells frame to frame, and it's going to flag for us the division events. But even with the most standard pipeline in the community, still tracking is challenged. And tracking is challenging by nature because it's very challenging to be able to track each individual cell over time for 72 hours, let's say. So we address this need in the community by developing post-processing software. We call it Track Refiner, and basically this is a specialized software only on tracking, and it basically increases the accuracy of tracking by increasing the lineage continuity. Okay, to kind of remind everyone, our goal from the beginning was to calibrate our model. What does it mean? We wanted to have a model that matches the observation as closely as possible. But in biological setup, this is unrealistic. Instead, and the reason for that, basically, as other people said in this room, is because of the stochasticity in biology, in initial condition, in measurement noise, and so on. But instead, what we can aim for is statistical similarity. And this is where our pattern or summary statistic comes in. And this is very interesting and very exciting because basically these patterns or these summary statistics are a very effective way to basically compress our rich spatial temporal data to a set of measurable feature. And they can range from basically fine-grained features such as, for example, anisotropy to high-level information such as aspect ratio of a colony. Okay, when you have your summary statistics ready, you can ask this question that what parameters in your agent-based model give rise to observed pattern? And to answer this question, basically you can go for two different setups. You can start with sensitivity analysis. And in this specific study that I'm showing in this slide, we had two parameters because we were interested in calibrating biophysical parameters of our agent-based model in gene. So the output of sensitivity analysis hopefully is understanding which summary stats are informative and where in your parameter space you have more chance to get realistic behavior. And then you can take the knowledge from sensitivity analysis to your approximation Bayesian computation framework. And basically the idea, the output of agent ABC or approximation Bayesian computation is to kind of understand which combination of parameters is giving rise to basically observed pattern. And what is ABC? Very short, ABC is an iterative process. And you always start with a prior, and prior basically is what you're looking at in the right-hand side, is a broad parameter space, is a broad distribution of parameter space. Now you are looking at the formation of posterior in each iteration. As you can see early on, the pattern is scattered, but as we iterate, the more tighter posterior or better estimation is popping up. Okay, now what? Now we are extending this pipeline to more complex system. Here you are looking at our synthetic consortia. We have two bacteria. One of them is engineered to produce toxin. This toxin called colicin. We can talk about the biology if you are interested. But this toxin is responsible for inhibiting the growth of the susceptible. So here in this basic experiment, fluorescent, like red labeled bacteria are attackers, those ones that are producing the toxin, and the yellow ones are the susceptible ones. And we couple this experiment to our agent-based model, and we couple our agent-based model to diffusion reaction equation. Okay, to close, I want to highlight that our pipeline streamlines the model calibration against microscopy data back in time. Calibration often relied on manual fitting. You would fit the parameters as much as your output looked reasonable. That was subjective, rarely reproducible, and time-consuming. Then the community shift towards non-spatial calibration, such as matching time series with your average in your population level measurement. But what we are pitching today is a fully integrated, spatially resolved framework. And we believe this is a critical step towards predictive engineering of microbial communities. And of course, you can't go far without a great team. I'm grateful to my advisor, and a special shout out to all of my talented undergrads that I have the privilege of training them over my PhD. And I'm also thankful to funding. And with that, I'm happy to take questions, concerns, or feedbacks.
Great. Very interesting talk. Thank you for your presentation. I wanted to ask, basically, to get questions together. First is, what is the features that you compute the posterior distributions on, and how many samples do you use to compare one parameter set?
Okay, I'm happy that someone asked this question. So basically, in this specific study that I showed, in this specific study, so basically the idea, like what our ABC does in this specific study is basically running ABM and calculating all of those summary stats that you can see here, like density, aspect ratio, and 4 other mores, and basically calculate the same summary stat for the observation, and basically compare these two, and if they are close enough, accept it, and if it's not, reject it, and it reiterates. And the number of samples, I have brain fog, but I think it was maybe 50 simulation, but the paper is online. Yeah, I think it was 50.
Do you know how long it runs for 50 simulations?
That's a great question. So all of these was run in Compute Canada. So it's a cluster that we have access to. So I don't know the information of the GPU and all of these basically technicality, but it's written in the paper. But I believe it took us 48 hours to put together all of these.
Thank you very much.
Of course.
This was a really very nice talk. Thank you. I was most fascinated by these nice visual representations of these laboratory results. So I was wondering about a kind of a different direction, how do you think about visualizations of computational results, and how can we maybe also make, especially for your research, what these models predict more appealing to the people outside of the field? Or can we maybe, I think there's kind of potential for outreach. Have you thought about that? Or do you have good ideas maybe? Or I'm sure you have, so I'm very interested to hear.
Okay, thank you so much for the question. So basically, in terms of, I should acknowledge that, yeah, it's very lovely to see, for example, those colorful time-lapse, like these ones, these colorful time-lapse microscopy output. Basically, in terms of how we visualize that, for example, if we give a talk to high schoolers or undergrads, or when we kind of want to ask them to join our lab, basically we don't change this. We just show them as raw as possible, because we want them to understand if they accept to join us, they have to analyze this. And this is, you can see that after, for example, towards the last time points, it's getting very dense. So the task of segmentation and tracking is going to be so challenging. So because we want to be transparent about the labor that we're going to ask them, so we show the time-lapse as raw as possible. But in terms of how we visualize the output of the ABM, so the software we are using is developed by Tim Raj and his team back in 2012, and it's called Cell Modeler. And for example, Kevin Foster's group in Oxford are using the same software. I guess I haven't seen many other groups that use the same software, maybe because it's very niche. But the software itself has a very good visualizing setup. For example, I haven't changed anything in this simulation. I guess this is directly from the Cell Modeler. But yeah, for the community outreach, they're very interesting, especially if you want to pitch to community that, okay, it's important to fund us because we are capturing things at the single cell level, and hopefully, and because, for example, in terms of the application I said in the very first slides, this is exactly, for example, if I want to apply for a scholarship, I definitely put lots of those raw images in that. I hope that I answered your question. Okay, fantastic.
Okay, thank you again.
Of course.